So in the energy minimization method, what we were discussing, we have got different concepts and terminology which are applicable to this. We have already discussed yesterday. And the goal is to reduce the energy to the lower level so that the conformation of the interactions, conformation that is fitting to the interactions, that is going to work in the nearby future. So in this case, we have got these energy minimization techniques. We are going to see for the type of the docking, like a rigid docking and the flexible docking. That flexible docking is the induced fit docking. We have already seen. Now, it is very important. This molecular docking is very important in the rational design of the drug. We have already seen the rational means it is based on the logic and science based, not by the trial and error method and vague manner. So we have got a scientific backup for what we are doing and what we are getting. That is evidence based. So in this particular case, what we are doing, we are identifying the identification of the ligand correct binding geometry. So this is correct binding geometry or a pose in the binding site. And why it is very important? Because the receptor has got a multiple grooves and it is not like a straight or a flat. Many times it will be having a different geometry. We have to fit the ligand in that. So we have to see the correct binding geometry where it is going to fit in the receptor site. We need to find so that we can have the prediction of the binding affinity and scoring function. For that purpose, we have to reduce the molecule, because when we are taking a molecule in the computer's simulated systems, if the molecule is under the high energy states, under the stress, where the benzene ring and aromatic ring is present in the flat manner, but what we are presenting in the docking, the molecular structure containing the aromatic rings, they are having a not a flat, they are bending. And in the molecular structure of aromatics, we are having a benzene ring will have a bond length between each carbon and another carbon is same. But when we are presenting in the docking, the molecule is containing aromatic rings. The bond length is varying from the one carbon to another carbon to carbon, it is varying. It means it is in the high energy states, so it will not work further. So we have to bring back to low energy states so that the benzene ring should be made into a flat, so that we can make the success of a correct scores. That is what we are doing in the minimization techniques. Say in this particular case, we are having a docking between protein and ligand. What you have seen in the molecular docking in the figure in this particular screen. So what you are seeing that is protein and the ligand. The ligand is very small in nature. Protein is very large structure. So it has got a pocket and it is perfectly fitting there. That's what we are going to find. But our molecular docking is not restricted to this. We have to see the docking a protein with protein. Say insulin receptor is a protein, insulin is also protein, where our response in the treatment of the disease of diabetes, where the receptor and the drug, that is protein, that is insulin binding, is also we need to see in the docking. Such cases, our receptor is also protein and drug is also protein. So nearby future, we are going to have this type of phenomena very high. More drugs and more drugs are going to come in the nearby future with which are having a protein in nature. So this is also a part in the docking. Another way, we are going to have a shutting the genes and modulating our responses with respect to the genetic. We are going to have a interaction between the protein and nucleotide sequences also, where RNA fragments, DNA fragments, where protein is required to be interacted to shut on the gene and shut down the gene, and where we are operating to reduce the disease conditions and treat the diseases. So these three types of the docking we are going to have. In this figure, what you are seeing is a protein ligand docking. What are the softwares available? Because we need to memorize where, as and when the topic is coming, we need to handle the softwares, tools, and the databases with respect to that. Whereas the softwares, when the national software, when you see it, one software is called as a Sanjeevani, that is developed by the IIT Delhi. So the software used for the docking in the national wide, and this is a home domestic software which is available. Another software internationally which is available is Gold. That is it is made by the University of Cambridge, UK. And another popular software for the docking is Autodock. So this is Autodock is developed by the America. So another dock is a GEMDock, Genetic Evolutionary Method for Molecular Docking. It is a tool developed by the Jinmoon Yang. So this is he is a professor of Institute of Informatics. So this is Taiwan-based software. Hex protein docking. So it is a UK-based, and GRAM, that is Global Range Molecular Matching Protein Docking Tool. So that is developed in the Center for Bioinformatics, University of Kansas, America. So these softwares are more popular with respect to our national, domestic, and international, that is global docking softwares. And there are so many advances are there in our agentic AI, what we are discussing to develop in computer-aided drug design. This software will become one agent in that. In the orchestrator, we are attaching this one as a one agent. So the software tools which we need to adopt, that we need to have a subscription and payment already ready, then we can use them for our purposes as a agentic AI part. So we can make them automated so that we will be making processing in a single stretch. Now coming to the energy minimization. So whatever the software we program we use, that is used to create the 3D structure from the that is single line structure, that is smile you have got, and two-dimensional structure like a mole structure where you have got a structure represented by the matrix form, that is length and breadth. And in the 3D structure where we are representing, you have got a length and breadth and the height. So that is having a to and fro depression also, top also. So you have got a three-dimensional structure where just like a cube, you are taking a line, you are taking a square, and you are taking as a cube. So this is what we are going to have in the 3D structure, where a process called as the energy minimization should be carried out. Once the structure is built, then only we will get the success. Say, for example, with the molecule what we have selected previously as a pharmacophore from the hit and lead modifications, then we have identified the pharmacophore and we are bringing it to a drug-like candidate in the docking studies. So this is appearing to be successful when the drug is having a highest energy. Then it is not going to work, because in the real state, the molecule will be present in the least energy only. It will not be distorted in the natural cases. It will be free floating in our blood and plasma in our body. So it is not in the high energy states. So we have to bring back to low energy state before docking. That is criteria we need to take. This is because the construction process may have resulted in the unfavorable bond length, what we have done previously in the two-dimensional structures, or maybe a software related problem, or what we have given the commands or the prompts that has made this unfavorable bond lengths and bond angles or the torsions in the angles. So they are different. That is the reason it has gone to the high energy states. So the energy minimization process is usually carried out by a molecules mechanics program, which calculate the energy of a starting material. It is having some energy state. Then vary the bond length and the torsion angles to create a new structure that is having a minimum energy without modifying basic atoms in the molecule. That is one type of making an energy minimization. So even you are not getting minimum energy with this, and after making the minimum energy also, you are not able to fit in the pocket of a receptor, then you need to make a changes in the functional groups. That is having a changes in the bond lengths, or it is further increasing by adding some homologous series, or we want to add some functional groups that is going to fit. Then we will be taking that moiety. Then we are making a pharmacophore identification based on the QSCR. That is quantitative structure activity relationship. We need to select another functional group or a different molecule. This is a reiterated, it is a iteration process. We are doing and repeating till we get the best. This part we are doing in the computer. So we are not having any expenses in the lab by synthesizing it, and really in that is a laboratory, we are having this is synthesizing and screening in the real state. But it is we are doing in the virtually we are doing the screening process. So the failure, we will detect it and we go to another molecule immediately. We don't proceed in that. And we are making the minimum energy states. That is the reason it is simulated to our real systems. That is the reason we are going to get a success rates very high. So the energy of the new structure is calculated to see whether it is energetically more stable or not. Because high energy systems are not stable. Any system wants to come to ground state when it is going to work. That is the reason we have to work in the minimum energy states only in our docking studies. So most of the times the reduction in the energy is not like a linear graphs. It is like a curvilinear graphs. We may not have the linear graphs. Most of the times it is curvilinear graphs, what you are seeing in this particular diagram. So if the starting structure is inherently unstable and a slight alteration in the bind angle and the bond length have a large effect in the overall energy of the molecule, resulting in a high energy difference, that is delta E. Then we need to make a modification in the tools in such a way that we are adjusting the bond lengths and bond angles so that it will be having to the really stable structures. Then we are fitting that particular moiety, fixing it, that three-dimensional structure of that particular moiety, we will fix it. Once we are fixing it, then we can see the score of interaction at the binding sites. This is our journey. So you see the figure where you have got y-axis is the delta E, that is change in the energy with respect to variation in the bond angles and your bond lengths. So what you are doing, variation in the bond lengths and the angles when you are making it. So the energy of unstable system, which is going to bring down to the stable system, that is the energy minimum system, that is represented as a dark black circle. That is the lowest energy state. So when we are varying in the x-axis different bond lengths and different angles, and we are going to have a so many responses out of it. So the we are going to keep on repeating it. After particular repetitions, when you are doing it, again the energy is, states is going to high. It means what we are doing modifications in the structure, molecular structure, molecular mechanics, that is going to increase again energy. We need to stop the reiteration process and see the what is the energy minimum states there. So that is what is represented as a closed circle, where we are starting is an open circle, that is gray circle, which is represented in this particular diagram, which is having a high energy states. So this is not like a straightforward, we are coming from the high energy state to and low energy state directly. We are going to do in a stepwise manner. So we are going to have a steepest descent method. We will be having a descending in a step manner. That is the reason we are going to have a curvilinear relationship here. So this is a reality. It will come like that only directly. When you are going to one place to another place linearly, what will happen? One system to another system, it may come down. But when you are not checking fully, then what will happen? There may be energy minima may be present, and somewhere changes we are missing it. That is the reason we have to reiteration process. We have to do repeatedly, and it will be keep on decreasing. We have to track that. And when it is increasing again, we have to track that and fix the energy minimum level in the structure. The structure, what is having an energy minimum, we have to pick up. That after picking up, we have to go for a docking. So this is the first step in the docking studies. So the program will recognize this and carried out more changes. Recognizing these changes, which lead to the stabilization, which do those which do not having a stability previously in the starting. Eventually, a structure will be found where structural variation result only a slight changes in the energy, slight energy minimum. So where we are going to find out where you will have a plateau stage also, we have to find that and fix it as a minimum energy states. That molecular structure where we are having this minimum energy state of a ligand, we have to take that further consideration. This is what our journey. We will see now an example. 16 persons are present in the room. Attendance purpose, I have taken already screenshot. We'll proceed now. So in this particular diagram, what you are seeing is the energy minimizing for the apomorphine. So the two-dimensional structure of the apomorphine you see in the first catechol ring, it is a two-dimensional structure. So that is having a two hydroxyl group, one nitrogen group is attached with one methyl group. So this is not generally flat in nature, because it is in the reality, the catechol ring will have some of the functional groups present below the plane of paper and some are present above the plane of paper. It is not like a two-dimensional, it is having a depth also and a height also. So it is having a third dimension here. So what we have represented catechol ring, we need to bring to three-dimensional space where it is aligning some of the functional groups below and above the paper. That is flat, what we have seen. That is what we are observed in the computer before minimization. This structure was presented. That is middle structure, where we are representing gray balls as a carbons and dark balls as a oxygen and black balls as a nitrogen. So one nitrogen is present. We are representing in the three-dimensional ball and string representation. What we have seen the structure, which is presented as a three-dimensional structure. So the computer has presented this structure to you. So what you have observed in that particular before minimization, what you have observed is the catechol is containing two benzene rings in the apomorphine. So extreme left, the one benzene ring is containing two hydroxyl groups. The software is shown the before minimization, the benzene ring is distorted. So it has got some saddle points where it has got a peaks and valleys when it is a flat. Really the benzene ring required to be a flat in nature. Then only we can fit each bond angle between carbon as same. But if you see it, the first hydroxyl group which is attached, the bond angle is very less, whereas another one is very high. So benzene ring really coquille structure will not have like that. So it is having a same bond angles. So once you found in the structure, the bond angle, once we have found is 1.42 angstrom units, another bond angle is 1.57 angstrom units. This is a highly energized state. It will not exist. We have to bring it back to low energy state so that we are going to come to reality, come to the closeness to the reality. So we have made the ring as a planar aromatic ring and represented the remaining automatically remaining structures, what is present below and above after making as a plane. So the two benzene rings after making as a plane, now you see all the band angles between carbon atoms in this benzene ring is same. So the planar aromatic ring is representing after minimization. So the energy state of this particular structure is less now. So this energy minimization carried out on the epimorphin. If you take the third structure, what you are seeing in this figure, it is going to work for us. If you go with a second structure, even though you get the success in the docking, it will not helpful because such a type of apomorphin will not exist in body. And when you are preparing your ligand in the system like this with high energy states, it will not present in the reality where you are getting a good docking stores also. It will not work in the market or when you are subsequently you are doing in the animal studies and clinical trials. So we have to bring back to low energy states. After minimization, we have to freeze that particular moiety structure and we have to take this structure for the docking, molecular docking with the protein receptor binding. That is what our journey is. So what is the energy minimization in short? It is a procedure that attempts to minimize the potential energy of the system to a possible lowest point. Because what is the one which you can attempt now? You have to bring back to very low only. We cannot make it to zero. So that is our aim is to see the maximum energy to energy maxima to energy minima. And you need to find out the packs again going to reiterations. It is not coming down again, it is going up. Sometimes you may have the plateau stage, but sometimes you will be going to again up. It means you are freezing a point because it is a time wasting to reduce further. Your reiterations, whatever it is showing the energy minima, you will freeze that point and you go ahead. Otherwise, it is highly time consuming process if you are going to away from the reality to ideality. Our aim is to manage our work in the real nature. No need to be ideally like this, because it is no molecule will be present like that, because it has got obviously in the structure because of its internal structure differences and the attractions and repulsions, it will have some energy states. So that we cannot restrict, because of that point we have to, our aim is to concentrate to the minimum energy. That is, in short, we are doing it now. So the system makes several changes in the atom positions and through the rotations and calculate the energy in every position. And it is iteration will be keep on going by the software tools. This process is repeated many times to find a position with the lowest energy until overall minimum energy is attained. So when you are having a locally in a particular atom, it is decreasing some energy, and ultimately we are going to have the impact in the global molecule. So the same distortion, it will alter the remaining distortions in the another functional groups also. So we have to see the local minima, what is its impact on the global minima. So there are two things. Local means particularly changing one atom, its bond length and its angles. And we are seeing the what is the local minima there. And it is going to impact in the global also, that is the entire molecule also. We have to see both. So global minima and even the simple local minima is not sufficient to us. We have to see the global minima energy state where we have got a molecular mechanics where we are going to work with a global minima also less. So the overall minima energy is attained. That's what we are going to see in short. So the one full round of atomic rotation is called as a minimization step. One full round. So we are having a 360 angle rotation is called as a minimization step or a one iteration. So you are making the atoms rotated like this. Each and every atom in the locally, we are going to rotate and find out the energy minimum states. And we are bringing all the atoms locally to the energy minimized states by iterations and bring down the energy state to the lower level and freeze those points and find the global minima. What is the global minima at that particular state? So what is the energy state at that particular one at the molecular level? That's what we have to see. For that purpose, we have to have a energy minimization for one is locating a stable conformation. So where it is having a lower energy and locating a global and local minima. So when you are distorting it, each atom, a particular point where you have got a local minima and global minima, both are under the lower levels. So how to do it? We have to locate the saddle points. What is the saddle points? Where you have got a highest distortions, which will be having a maximized states, highest maximized steepest states. In the diagram, what you have seen, the curvilinear relationship, it is appearing like a U. So, but in the saddle point, the energy is so high, it is appearing like a mountain. It is reverse of what you have got a U diagram in the previous case. This is what I am going back. You see here, in this diagram, you have got energy minimum is U. That is minimum energy state in the dark circle, what is present. But in the saddle point, what will happen? It is a highly distorted state. And the energy changed to the highest point there, where this U, that is valley, will become like a mountain peak. So this in that particular saddle point stage, this circle will go to top and it will be going to represent a high energy states. So this is the worst point in our scenario. This is worst point. So sometimes what will happen? We are starting our journey at the saddle points only. So the saddle points, we are having a reiterations many times, locating the global and local minima. And after fixing the global and local minima, then we are going to have a locating a stable conformation. This stable conformation, we will freeze it and use it for the docking studies so that we will get a good scores. That is going to be a successful. I hope you got the point now. There are 4 steps in this journey. One is computation of the potential energy of the static geometry, where we are starting with a one structure. So we have to do that. So we have got one structure, we should not proceed it. With that particular structure, we should not go for the docking in the receptor because it is time consuming process and you may fail. That is the reason we don't want to do it. First, we need to compute the potential energy of the static geometry. This is workflow. So the second step is alterations of the atomic positions of each atom and computing the energy of entire molecule. So we are altering it in a 360 angles and computing the energy states of molecules, entire molecule. So that is global minima and the local minima. Both we are seeing it. This entire molecule, what is the energy states also, we are finding it. If the energy of the third step is, if the energy of new conformer is less than the starting one, adopt this conformation and proceed for the next alteration. Otherwise, retain the first one. So say for example, you are starting with one energy state. When you are changing, it is going to the saddle point. Then you don't want to, otherwise you have to go back original position only, because it is reaching, the distortions are increasing the energy states. So it means it is going to saddle points. So in the saddle points, when it is going, you have to come back to original point that is having a energy state. So that is better state. In fact, we are making a changes and reiterations that is worsening our case. So we have to repeat the process. That is the fourth step. Still, there is no more decrease in the potential energy. We have keep on doing it, and there is no more decrease. When we are doing this particular process, there is no more decrease. So we are doing like this. We are going to find a minimum, and in that journey, we are finding a saddle points and maximum energy maximums, and we are coming back to previous original states, and again we are making the reiterations, and we are coming to a decrease in the potential energy states. That is called as a energy minima. This is what very, very important in the computer aided drug design. This is what the computer is doing. We are not able to do as a human being, because human being with a naked eye, you cannot see the protein as an isolated moiety. You cannot see the receptor as isolated receptor cavity, because it is having so many cavities, and the ligand, it is invisible. So all are invisible, and how to find out when it is fitting or not? Now you may get the question, sir, I am having a receptor and the ligand. I am going to put it in a reaction vessel and both are combining them. Later onwards, the complex I will take it. And even though it is not to my visible eye, I take that particular receptor ligand complex and cryo electron microscopy, I want to do it, because electron microscopy, it will be revealing the what is not visible to the human eye, it is revealing, it is visible. So I am that is freeze drying it, because I am not going to have any distortion in the freeze drying. Just we are removing the water molecule and making it a dry state. And we are going to a that is microscope, electron microscopy, where I am going to see the structure where it is having a contact points. Sir, where I have got the contact point, 4 places I have got ligand and the contact points are present in the cryo the electron microscopy study. So what is that contact point? How will I know it? Whether it is nitrogen or carbon or it is sulfur or oxygen? How will I know it? Then I go to NMR. So the NMR reveals where the contact point is having linkage between carbon and nitrogen and sulfur. What the linkage is present, the NMR will tell us. the structure will be telling it. So what is not visible to me, it is visible now. Similarly, I can see the internal volume of that particular pocket. What is the space which is present in the packet by the XRD, where I am having a X-ray diffraction techniques, where it is having, there is a gaps which are present, a diffraction to the bending of the light, wherever the molecule atoms are present, which is bending to the geometric, that is bending of a light in the geometric shadow. So I am getting a XRD patterns, so that I will be able to find out the what is really internal structure is. So by this, even though I am not able to human eye, I am making it visualized in the particular case. This is all the called as pragmatic approach. That is what you are going in the lab. So, but it is a highly cumbersome process for doing like this, isolating it. During the isolation, your complex should be retained. Then you need to freeze dry it. So costly affairs and time consuming efforts. So these are all things we are doing it. And you are uploading the particular structure where which one of the amino acid, which is got a linkage with what atom of your ligand, then we are able to find out. That's what we were doing previously. Once we have done this computer software and AlphaFold recently in 2020, this is all the work done by the software. So this docking and the energy minimization and your AlphaFold is giving the structure of your protein and pockets. Based on that, you can calculate your geometry. Everything is now possible in the computer. So why can't we do it? So that is the reason we were here to learn all these concepts so that we can avoid our adventures and failures in the lab state. And we are going to finalize our moiety before entering into the lab. This is what our journey is. So the minimization is to find a set of coordinates representing the minimum energy conformation for a given structure. That is what we are aiming at. And we are avoiding all the lab tests. That is what you are getting in the pragmatic manner. That is taking a time. So we are going to use the virtual screening so that we will not have all these steps. Various algorithms have been formulated by varying use of the derivatives. So some algorithms are based on the first order derivatives. Some algorithms and softwares are based on the second order derivatives. First order derivative means the change in the structure, that is the atoms bond angles and bond lengths changing in the energy, that is delta. So, and we are going to second orders. Changes in this and how much is the changes we are going to have further. That is second order, third order. So this is actually what we are going, the various algorithm and software tools, they are having different techniques. So there are three common algorithms are used for this particular optimization and iteration process to find out the low energy states. One is steepest descent method. Steepest descent method, we are gradually descending to one. That is the lowest one which we are finding. In our optimization also, we are going to the steepest ascent and steepest descent method. Say, for example, in our formulation development, we want to use a software and we want to find the conditions where we want to increase the dissolution rate. That is the dissolution percent. We have to find out the conditions of our formulation parameters where we need to have the maximization of our release, because that is called as the steepest ascent method. But when you want to have a sustained release, where you want to decrease the dissolution, then you need to use the steep and descent method. So you want to decrease your disintegration time, then you are going to use a steep and descent method. So where you want to increase the what you have got hardness of a tablet, then it is steep and descent method. So depending upon what our requirement, here in this particular algorithm, we are going to energy minima. That is the reason we are going to use a steep and descent method. Similar concept we are going to use in drug discovery also. Same concept which are used in the software of our formulation evaluation also, where we are taking parameters for the steepest ascent, sometimes steepest descent sometimes. The second method is conjugate gradient method. The third method is Newton-Raphson method. So Newton-Raphson method. So other methods are also available. That is downhill simplex method. Our reiterations, we are going with a 1 1 1 step down, and you are going up to down. In the simplex ascent or descent, there also you can target to the what 1 change as a trial and error. We are going to increase or decrease, we are going to see and take the next step. Keep on doing multiple steps like this. That is actually called as a downhill simplex method. So these 4 methods we can consider generally being used, and there are so many software versions, they are doing so many advances also. This will be keep on changing day by day. So what is available now, we are going to see one by one. In the steepest descent method, you are seeing on the diagram. Now we have started with potential energy, that is the energy total of the system in the y-axis. In the apex, you have got a gray ball. So in the gray ball, when you are having variations in the parameters like atomic bond lengths and atomic bond angles, you have come to the local energy minimum, that is A. So it is slowly you are doing a reiterations from the starting point confirmation. You are doing a change in the potential energy. It is down, coming down, slowly coming down. So you are happy, but you have got a point A. This is the local energy minimum. So it is in that local energy minimum, you have done some more changes. But in the changes, when you are making it, again, it is going, potential energy going to the higher level. So it is against your requirement. Now again, peak is coming. That is, you are getting a saddle point now. But in fact, it is not a saddle point. It is lesser than the original point. That is the reason we don't call it as a saddle point. So if the B is going above your gray, then it is becoming a saddle point. It is minimum energy state only when compared to your starting point. Say, for example, if you have not done some iterations, you have done directly iterations of B, then you will directly go to from gray ball to B. So you will not have A. You might have missed it. That is the reason you have to reiterations carefully. You have to do it in all the angles, on possibilities, all possibilities. You have to do it. Don't miss any possibility, so that you are missing a correct minima by missing some possibility. So when you are getting an energy barrier, when you are doing those steps, when you have got an energy barrier in that one, again you are reiterating it and coming to the point C. So that C is represented as a global energy minimum here. So the global energy minimum is, again you are doing a modification in the reiterations in the variations in the parameter. It is going up again, reaching to A. So it means that at present what we have got in the tree A and B, the third dot is a global energy minimum, which we have found out with energy total is very low. So this is what we have arrived at. Simplest method for the geometric optimization of a ligand. It is also called as a gradient descent method. Optimization algorithm for obtaining a local minimum of a multi-dimensional function. So we are doing this. The energy minimization methodology needs to involve identification of the point closest to the starting structure. And we are going to find out the energy minimization, which is having a least one. We have to identify it. So we are seeing the differences from the starting, and we are going to reach to the global minimum, global energy minimum later. So this local energy, and it is creating a barrier, increasing, and you are making further changes in another atoms structures modifications, and again it is having a energy barrier high, and we are reaching a particular modifications repeatedly. We are doing it. Then the local minimums and going to a crossing energy barriers, we are going to a global minimum state, where this is the total moiety, which will be having a potential E total when accounting for all atoms in the molecule, in the molecular mechanics, which we are going to bring that particular state to the lower level. This is called as a steepened descent method, and we will see remaining concept tomorrow. So I will open the floor for discussions.